I'm going to show you how you're going to do your data analysis for the Density Lab. First, you're going to download a copy of the template, and it will look like this. You'll need to rename it with your name and Density Lab data table. And then you're going to enter your mass for your 4 trials and your volumes for your 4 trials, and it will end up looking something like this. Next, you're going to calculate the averages for your masses. To do calculations in Google Sheets, you're going to use formulas, and each formula starts with an equal sign. So you type equal, average, and it's going to suggest what it thinks you should be averaging. It highlights the cells, and indeed we want sample A, the 4 trials of them, to be averaged. So it's complete. All we have to do is hit enter, and there's the average. To do the averages for the other cells, I'm going to grab the blue square at the bottom right corner and drag it down, and it's now calculated the average for each of my trials. I'm going to repeat this for volume. It's already telling me the average. I want those 4 trials, so I hit enter, drag the blue square, and there are my averages. Let's take a look at our significant figures. Looking at the volumes, all of my volumes should have been to the 10th place, and all of my volumes are to the 10th place, so I don't need to adjust anything there. With the masses, however, my measurements, the balance, measured to the 100th place, but my average is only to the 10th place, so I need to fix that. So I'm going to select those cells, and then I'm going to use one of these 2 buttons. Either decrease or increase the decimal places. In this case, I need to increase the decimal place because I need to get to the 100th place. And now I have my averages to the 100th place. Now I need to transfer my average mass and my average volumes to this bottom data table. And I'm going to do that by linking cells. And the way you do that is you hit equals. For average mass for sample A, I click on that cell. And now those 2 cells are linked. So if I changed one of the numbers in my trial, that would change the average, and it would change the average down here. I use my blue box, drag that corner, do the same thing for volume, hit enter, drag my blue box, oops, let's try that again. And now I have my average mass and my average volume linked from these upper 2 data tables. Now I need to calculate my density. To do that, I need to divide average mass by average volume. Since I'm going to use a formula again, so I use an equal sign, and I select the average mass cell divided by the average volume cell, hit enter, and there's my density. Grab the blue box. Now I went a little too far. I don't want to do that yet. So now I have my densities calculated from the average mass and the average volume. Now you can see immediately that the significant figures are far too many, so I select all of those cells, go back up here to my decrease or increase decimal places. I need to decrease, and I need to decrease to 2 places from the average volume. So I'm going to just click on that until I'm down to 2 significant figures. So now this table is done. The last thing I need to do is I need to average all of my densities. So I select equals, average, it's selecting the correct cells. I hit enter, correct number of significant figures, and I'm done. I'm going to color this yellow. That's in the paint bucket, fill color. And And now I've done all of my calculations. The next thing I need to do is to graph this. To do that, I'm going to select my average mass and average volume cells. I go to Insert and Chart. Now it's given me a column chart. That's not what I want. So I'm going to go in the chart editor to a drop-down menu, and I'm going to go down here to Scatter Chart. Now I'm going to need to do some customizing. First, I need a title for my chart. So here's chart title. I'm going to type density of an unknown, and I'm going to describe the color. It was an opaque gray solid. So for you, if you had a black solid or a shiny silver solid, you would describe it here. Next, I'm going to label my axes. My horizontal axis is volume, and it's in milliliters. My vertical axis is mass in grams. And so now I have a title and my axes are correctly labeled. Next, I need a line through this graph, and I do that under Series, and I click Trend Line, and this gives me my line of best fit. I need the slope of this line. To do that, I go under Label, and under the drop-down menu, I select Use Equation, and now I have the equation for the line. This is in the form of y equals mx plus b. I don't care about the y-intercept. I care about the slope, which is 1.19. So I'm going to enter that down here, but remember I need two significant figures, not three. So I'm going to call it 1.2. And now I have my graph and my calculated average density and my density from the slope of the line. So I'm done with the data table. Now I need to link this chart to my my file for my lab report. So here's my docs page. I'm going to go Insert, Chart, from Sheets, select my graph, click on that, and it imports my graph. And the nice thing about this is that if I actually went back and changed things in my data, which changes my line, it would change my graph in my file also. So everything is linked. And this is how you do your density lab data analysis.